Hi guys, today we're going to be talking about what is dielectrophoresis. Now gosh, that's a pretty big word, so let's break it down into parts. Di stands for two, the prefix. Then the next one is the root word electro, which usually means electricity. And whenever we're talking about electricity, we're going to be talking about charges, which is an attribute of an object that's either negative or positive. And the general rule is that opposite charges attract and like charges repel. So the next root word is phoresis, which means the movement of something. So if we put together electro and phoresis, we get the movement of charges. But the question remains, where are these charges moving, and exactly what are there two of? Alright, so to answer that first question, we're going to have to talk more about charges. So remember how opposite charges attract? Well, how exactly does that work? Does the positive charge reach out a hand and physically bring in the negative charge? Not necessarily the case. This all happens over vacant space. So what happens is, each charge exerts an electric field, which are these invisible vectors that exert the attractive and repulsive forces between charged particles. So when we're talking about dielectrophoresis, the where is in an electric field created by two electrodes, which is just a collection of charges that creates an electric field. So here we have a positive and negative electrode. So let's put a neutral particle between them. This means that it has an equal number of positive and negative charges. So there's going to be an attraction this way towards the negative electrode and an attraction this way towards the positive one. However, since the electrodes are the same size and same shape, these two forces would cancel because they're equal. So in order to have actual movement, we're going to have two different electrodes, a non-uniform electric field. So now this circular positive electrode is going to have an electric field that goes all around like this, whereas the negative one is going to be weaker because it's only going in these directions. Therefore, there's going to be a net movement towards the positive electrode. So in this way, but by controlling the electrodes and the power of these electrodes and the shape of these electrodes, we can control the movement of the particle. So it turns out that dielectrophoresis is actually pretty important. It allows us to analyze and isolate cells. Well, why is that such a big deal? Well, let's look at this dielectrophoresis model here. All the cells that are migrating towards the electrodes are actually cancer cells, and the other ones are normal cells. So here, scientists are able to isolate cancer cells and give them specific treatment, instead of giving treatment to all of these cells and killing them off, which is better for the patient. In this way, this physical phenomenon gives us a better way of analyzing biological systems. Alright, so that's pretty much dielectrophoresis in a nutshell. Thank you so much for watching, and if you have any questions, please comment below. Bye!